forward real quick. Okay, so with this, a couple of quick things that I want to talk about for lab. One thing that I noticed a lot, and we didn't harp on the first week, but we're going to do it next week more, is if you have pant legs that go, you need to have pant legs go all the way down to where your shoes are. So if you like sit down and you have like a big gap between where your pant leg ends and your shoe, and you have a bunch of exposed skin, well, that's not a good set of proper lab attire, because if something's going to drop and break and spill in lab, it's going to happen probably on the floor. And if you have a bunch of exposed skin really close to the floor, that's not the place you want to have it. Dr. Lawson suggested that I show you guys a bunch of gross pictures of chemical burns on ankles, but I haven't found any, and I don't need to subject you guys to that. I'm sure you guys will all be willing to go look them up yourselves on the internet if you don't believe that that's a safety hazard. Okay, so we're going to talk about transformation and isolation. So the transformation and isolation lab, a couple of safety things. First off, make sure, of course, your work area is always decontaminated, disinfected, as well as wash your hands before you start. Do not carry any test tubes around by hand, always use them in the rack. The reason why test tubes are notoriously round and slick, and if you bump into people, the likelihood of you dropping them while holding them is really high, but if you have them in the rack, very hard to drop them. You can, it's just really hard to. If we have any slides, which you don't really have to worry about for this experiment, but if you do have a slide that breaks, let us know right away. Notify your TA of any spills that happen. Do not handle the back T-incinerator, so don't touch the outside metal part. It's hot, you can burn yourself. And the same, so just keep that in mind. Obviously have on goggles, gloves, and lab coat at all times in the lab. We will be using a BSL-2 agent, so make sure you guys keep that in mind. Now, transformation. So basically what is it? It is the process of horizontal gene transfer by which bacteria pick up genetic material from the environment. And I know what you guys are asking yourself. Is there such a thing as vertical gene transfer? And the answer is yes, but it's very difficult. And I won't go into it details right now, but basically all of it's horizontal, so kind of all across same species, same genus. It's not, don't worry about it too much. But it's not such a key thing when we're talking about bacteria. It's all considered horizontal for that part. So how does this work? Well, some bacteria are really, really, really good at just picking up DNA from the environment and saying, hey, you're my new friend now, and you're going to be part of me, and just using whatever that was on that DNA. This is how they pick up things like resistance to antibiotics, so on and so forth. So here's an example where DNA is taken up by competent bacteria. They are able to utilize it and then either create a plasmid out of it, so it's a circularized bit that's all distinct from its whole genome, or incorporate it into its genome. So there's, of course, the famous experiment regarding this with the mice. So they took they took virulent colonies, and they were like, hey, let's do some experiments right here. And so they said, okay, we've got this non-virulent rough type, R-type, bacteria. We'll inject them into mice. Mice alive. Okay, let's take some smooth type, which is the virulent type. Inject it into mice. Mice died. Let's take some smooth type and heat kill it. So put it under a lot of heat and inject it into mouse. Mouse lives. Okay, great. Heat kills that. Now let's mix R type and smooth type that's been heat killed. Mouse dies. That doesn't make sense. And this is literally what happened. They were kind of confused initially because they're like, that's not what we predicted would happen. And what we're seeing here is that perfect example. Because there's this DNA in the solution that the bacteria are mixed in from the dead bacteria and these completely competent rough type bacteria, they were able to pick up that gene that allows it to kill the mouse. So what do you know? And then when they looked in the microscope, they were like, hey, those bacteria are portraying S-type qualities. So there you go. So that's what transformation is in a nutshell. And we're going to talk about some other methods too. Now, we use this all the time in science, and the most common one that you'll see is putting some sort of label inside a cell. In this case, fluorescent labels. They're great. You can do a bunch of art with them. There's a whole bunch of different colors, and we will actually do this later on in the semester. So keep that in mind. We'll have some fun with these. This image is a little modified so they're brighter than they appear. in real life, but they do have these colors, and we have a good variety of them. So you guys will get a chance to play around with some colorful bacteria. But these are the ones that kind of describe which kind of genes we put in there and how we're making them, so on and so forth. So you guys will get an opportunity to play around with that, but that's an example of transformation. Now, there's some other ways to transform cells in a lab rather than just hoping it picks it up, because that's not a great way to do science, is to hope things happen. So we make things happen. One of them is with an electron gun. It does not look like that. It's far more boring, but I like to pretend that that's what it should look like. It looks a lot like this, where basically you are shooting DNA with a small puff of electrons, and you're basically hitting the DNA into some gold particles, and it just kind of shoots into the cell at high velocity, and they get stuck in there, and now the bacteria have DNA on them from your desired source. The benefit of this is gold is relatively good at, it's relatively easy to bind DNA to gold or get it to stick to gold, and gold is relatively non-toxic to living cells. So it's actually a really convenient method. But we have much more simpler methods currently, though we still employ this from time to time, especially with bacteria that are resistant to taking up DNA. So that's the reason why I brought it up. Okay, another one is electroporation. Basically, we take the cells, we put them in some salty water, we put the DNA in the salty water, and we shock them with some electricity, and the electricity causes the DNA to move through the cells, make them all wobbly, and help the DNA pass through it. Because DNA is charged, it'll go through the cells, and then you just turn it off at some point, and now the DNA is inside. It didn't move all the way through the cell. That's how it works. It's really simple, pretty easy to do. Only downside is that you have to do a whole bunch of high voltage, which isn't the best, and it sometimes kills the cells. So the last method, which is the one that we're going to be using, is bacterial transformation with chemicals. We can basically solubilize their membrane a little bit, so make them a little unstable, allow the DNA to pass through easily, and then get them to kind of go back to a nice, regular, healthy state, and then grow them, and now they have that DNA on the inside. Now, some will have the DNA, and some will not. And we're going to talk about how we figure out which ones do and which ones don't. You guys probably already know this, but we're going to go over it again. What this whole process, which we talked about, is transformation. Now, there's some other methods for getting DNA into cells that aren't transformation, and one of them is transduction. Now, normally the way you do transduction is that you have phages bring in DNA from an outside source, so they infect the cell, they grab some of the cell's DNA, and then they infect a new cell and dump some of that DNA, and that cell happens to live, and now it has new DNA that it didn't have before. So that's kind of an easy, short version of this. Now, these guys here, these researchers, were doing a really cool study. So this is our Cool Science Thing of the Week, by the way, in case you guys are wondering. They were like, hey, let's take some mixed cultures, because mixed cultures are how we see most of the environment, most of the world. And so we'll mix together some cultures of a type of susceptible bacteria, so it's susceptible to this type of phage, and some resistant bacteria, so it's resistant to that type of phage. We'll mix them together, and we'll see if we can get them to lyse next to each other, because one of the things the phages do is they produce enzymes, or they make the cell produce enzymes to lyse them, and so if you're really close to another one, you might be lysing them, the resistant ones as well, and so they were trying to study that thing. And so they started doing this, and to differentiate them really easily, one of the things they did is they made the resistant ones glow green, and then they made the phages produce a red fluorescent gene so that they would glow red. And what they started noticing when they were looking through the images, they were like, hey, there's some red stuff stuck to this green guy. Red stuff shouldn't be stuck to green guy, because green guy is resistant to red stuff. And they were like, why is it doing that? And so then they spent another year studying this, because they had no clue what was going on. So this research paper turned into be a way bigger one, just because they were paying attention and didn't just go, oh, we'll throw away that data. That's clearly just nonsense. We'll just focus on the stuff that does what we want it to do, more or less. They went into this and really studied it, and they found a really cool, not before seen, method of transduction, or transformation, depending on how you think about it. So I'll explain. What they were able to do is they were able to go, hey, what is going on here with this? How are these cells lysing? So this is what the lysing cells look like. What they were able to do is they were able to capture these little tiny micelles that were being produced And the micelles are little bits of the cell membrane that have circularized and kind of been their own little packet, and sometimes they'll have a receptor on them, and that receptor will sometimes be able to bind a phage. And the real interesting part is, occasionally, if a micelle bumps into a cell that's just any cell, it'll sometimes just kind of incorporate itself into the cell membrane. Well, now, because it has a phage stuck to it, now it can just go inside the cell. So these cells that are resistant to phages, because they don't produce the right kind of surface proteins that the phages can bind to, suddenly become able to be infected by them. And they repeated this experiment in many, many, many permutations. And they were able to consistently show that, yes, you can in fact infect these. They did some really cool stuff. I think I have a video here. This is a live transfection. So these are cells that are getting infected with phages that produce a fluorescent protein. And you can see them getting infected, they glow, and then they get to the point where they lyse. And so that's what real-time lysis looks like. Well, it's not real-time, it's sped up a little bit. But I think this is over a course of an hour. So you can see these cells basically getting infected by phages. The phage is starting to produce that protein that fluoresces along with the other proteins it needs to survive, and then causing these cells to glow, and then you can see them lyse in real time, which is really cool. So in case you guys are wondering what cell lysis looks like, that's what it looks like. Okay. Okay. Any questions on that? Makes sense? Yes, I know. Cool science thing of the week. Take home lesson. Small little micelles glom onto resistant bacteria and help them become now susceptible to phages. Okay. So what are we doing? We're going to be doing a heat shock protocol. The way this works is we are going to give you guys a set of plasmid and a set of bacteria, and we are going to have you guys transform the bacteria. These bacteria, you guys will take from a plate, you guys will mix them in both of these broths, right? So it's got calcium chloride in it, so you just swirl it around. You'll then take a sample from the positive and the negative and streak it onto a Loria broth plate and a Loria broth plate with ampicillin. Then you guys are going to take your plasmid and then your control, which is a negative control of water, and you're going to add the plasmid to the positive one and the water to the negative one, and you're just going to let it chill for about 15 minutes. Just hang out, room temperature. Well, actually, it's on ice. Just let it chill. And the reason why we do that is we're just letting the cells kind of get comfortable, letting everything mix evenly, getting the DNA to kind of stick to the outside of the cell. And then what we're going to do is we're going to torture them a little bit. We're going to bring them up to 42 degrees Celsius. Now, humans and most bacteria, especially these ones, like about 37 degrees Celsius. That's what body temperature is. And that's where they grow optimally at. 42 degrees Celsius, not the best temperature. Makes things stress out quite a bit. These bacteria can handle it for brief periods of time, which is why we're only doing it for 50 seconds. The calcium chloride and the heat helps them solubilize their membrane, so they kind of get a little loose, a little melty, and allow that DNA to pass through. Then we're going to bring it down on cold again on ice for 1 to 2 minutes. We're going to let it sit there and chill. And this gives the cells a chance to kind of regain, breathe, and kind of re-solidify themselves. Then we will take some 37 degrees Celsius broths. Remember, this is the temperature that they like to grow at. We're going to add them to these tubes for 10 minutes and let them chill in an incubator at 37 degrees Celsius. This gives the cells nutrients to kind of repair things, kind of get set back up, start replicating a little bit. And then we're going to take a sample of them and we're going to transfer them to Loria broth plates and ampicillin Loria broth plates. And we'll streak them in this pattern here and we'll grow them. Now, what's the plasmid contain? That's a very good question. So our bacteria in this case are bacteria that are sensitive to ampicillin. So if you give them ampicillin, they will die, and they are not happy. So we work around this. We select for the ones that have the plasmid by giving them a plasmid, transforming them, and that prevents ampicillin from killing the cells. Now the way this works, really simple, really easy. Okay, not really simple, but they've got a little gene on them called ampicillin resistance. It's going to produce an enzyme, and this enzyme will essentially go to the ampicillin and cleave it right at this 4-ring corner, right there. and basically degrade it. So now you don't have that ampicillin around anymore. It's not destroying your cell membrane, so the cell is able to live. So if you have ampicillin resistance, you don't care if there's ampicillin there. You can just live without it. Now the next part, this is where it gets tricky. One of the things that I did not tell you is that these plates also have arabinose on them. And we have this control ring right here, Ara-C. Now, what does this do? Well, Ara-C, which is a really poorly named protein, but there's Ara A, B, C, and D. C, though, as the name implies, is a controller. So when you produce this protein, so it makes an mRNA, it goes off, becomes a protein, and that protein is going to bind to this promoter region called PBAD. BAD, arabinose, A, B, and D. Now, when it's there, as you guys saw right there, I'm going to back it up a little bit, it will not allow any kind of replication to go happening, transcription to happen. It's just going to block that off. It's very, very good at that. Good at shutting it down, making nothing happen. But in the presence of arabinose, it will pop off of there, and now it'll be free. So if there's plenty of arabinose around, it'll let go. So normally this is controlling arabinose metabolism in cells. Obviously we don't care about arabinose metabolism, we just care about the control mechanism. That's why we're using these guys. So it kicks off the arabinose Ara-C, and then it'll allow it to start transcribing the GFP protein, which is our fluorescent protein, which does a nice little glow. And there you go. Now, what does this look like? Well, this is a plate we did from the summer. So these are bacteria that are growing on a plate that's just Loria broth. And so the just Loria broth plate that doesn't have any ampicillin on it, both bacteria grow. So this side grows, they both grow on there. Then the one with ampicillin and arabinose on it, the ones with arabinose are starting to produce fluorescent proteins, and they will glow, and they only glow on one side. Now, I know you guys see some right over here. I'm going to stress this really carefully. If you guys are just teeny bit lazy with aseptic technique, a little bit of splatter, a little bit of splash can get over there, and that's why we have those colonies over there. So those colonies are kind of only on the edge. You got to be very careful with that. These guys more or less did a good job because this was the first time they were doing it. But you can see that most of there's basically no bacteria here, and the only bacteria that are here are the glowing ones, the ones that are able to survive under ampicillin and produce the nice glow. By the way, just so you guys know, if we took away Ara-C, we wouldn't have, if we took away the arabinose, it would not glow. It would just grow on the plate, just with ampicillin. So we have to add the arabinose to make it glow. If you don't have any arabinose, no glowy. It just survives the ampicillin. Okay, any questions on that? I know that was kind of confusing and a lot of stuff to throw at you guys, but that's what it is. Now, what we will be doing in this little, so many slides. Okay, I should have put another slide in there. Right in this little area is this area right here, which is kind of a big lag time. It can go longer than 10 minutes. 10 minutes is like the minimum time. You can let it go for like half an hour. In this time frame, we will start working on the isolation of a mixed sample, and you guys will be putting your streak plate knowledge to the test. So this is kind of where we're cramming those two labs in together. So you've got actually plenty of downtime in here, and you guys can take your time making your plates, because this is slow, it doesn't matter, we're not in a rush right here. Oh, so many slides. Why did I do this to myself? Okay, so pure isolation of pure cultures. Now remember, we talked about in the environment, most cultures are mixed. And so when you're sampling organisms from, say, wounds or from soil samples or water samples, there's a bunch of other stuff in there, and we don't necessarily always want that. So we try to get rid of that. And the way we do that is through subculturing. Now, if you guys remember, so these are little examples of subcultures, which is not exactly what bacteria have, but they do actually go through a small emo phase as they grow up, so just keep that in mind. What subculturing is for bacteria, process of transferring bacteria from one media to another to maintain an active growing state in order to perform biological tests. So that's all subculturing is. Now, within this, we can also streak for isolation, which is a type of subculturing. And this helps us ensure that we have a single genus and species. Most important thing. So every time you see one colony, single genus and species. Remember that. We just talked about this last week. We're going to do it again this week with slightly higher stakes. In this case, from last week, we gave you a pure culture. This week, we're going to give you a mixed culture. And the mixed culture is going to have Serratia marcescens and E. coli, which you guys worked with. The Serratia makes a nice bright red color. The E. coli is just a normal tan color. So they're easy to differentiate just on color, but we're going to give you them so they to analyze their cultural characteristics, which I'll talk more about in a second. So real quick, real brief reminder for how to do a streak plate. We always label the bottom of plates, never the lids. Just draw on the bottom of the plates. You don't have to put tape or anything on there. Draw a line down the center, line across. You're going to lift the lid up. You're going to have the plate, this end of the plate, against your palm. You lift the lid up slightly. You'll take your mixed culture, so you'll decontaminate your loop, grab your mixed culture, and streak it up and down here. Biggest tip, and this is the thing that people forget the most, so I would note this. Give your mixed culture a little swirl so that it's evenly distributed. Some bacteria like to sit at the bottom, some like to sit on the top. You never know. So if you have an even distribution, you're likely to catch both bacteria. In this case, we want both that bacteria just isolated. So keep that in mind. Close the lid, flame your loop. Take it out, let it cool down for a second. Turn this 90 degrees. You're going to lift it up again so that your palm is on this side. You're going to streak across the line. 1, 2, 3, 4. Close the lid, flame your loop again, let it cool down. Turn one more time so that your palm is right here. Lift the lid up and drag across here, up and down 3 or 4 times. This should give you guys at least one isolate of one type of bacteria. More than likely, though, you guys will be able to get more than one. So what we'll do is we'll give you a mixed broth. Everybody's going to have a shared mixed broth, so please don't contaminate them because it'll ruin it for your lab partners if you do. Don't be mean to your lab partners. Once you guys are done doing your inoculation, you guys are going to tape your plates together as a lab group, and you're going to put them lid side down in the room temperature incubator, which is the bluish incubator in the back. The reason why is because Serratia marcescens typically only does its pigment at room temperature, and it doesn't really like to do it at high temperatures. It will sometimes. We have a special variety that likes to do that at high temperatures. So it'll definitely be very red for you guys no matter what. The other benefit of this is for the inoculation, at room temperature, everything grows slower because it has less warmth to grow. And basically, the growth rate is dependent on factors like temperature. So if it's colder, just like us, bacteria don't like to go out when it's cold, neither do we. So they just grow a lot slower at lower temperatures. So you can get smaller colonies, which is very helpful if you're trying to isolate them. So we'll just kind of grow it at a slower temperature. We will look at these not on Wednesday, because we'll be done for that week. We will look at them next week on Monday, first thing, and look at our results. I will actually take them out of the room temperature incubator and put them in the fridge. This is where it gets really important. Label your plates really carefully, like right on the tape that you guys tape your group together in. So you guys should have 4 plates that you're taping and putting in that incubator. But section A, blue group. Section C, red group. Make it really clear so it's easy for you guys to find them because I want to take them all and put them in a biohazard fridge so that they stop growing and you don't have overgrowth. So you guys get isolated colonies. So how do we look at colony characteristics? Well, there's this chart on Lab Archives that you should be able to see. We're going to go over it briefly here. And there's lots of different factors you can look at. You can look at pigmentation. Some bacteria produce a nice light pink. So this is Cochuria rosea, which is a small tetrad-forming bacteria. It's got a nice light pink to it, peach-colored, some might say. Right here you can see a nice bright white. So this is Staphylococcus epidermidis. It makes a nice very white coloration to it. Some are tan. This is I believe this one is Pseudomonas aeruginosa, and you can see it's actually got a light green to it, but the bacteria themselves are tan. They produce a light green pigment. Aside from this, you guys can also see colony size. Colony size is really easy to differentiate. It's small. I know it's really hard to see in this image, but if you guys look really close on Lab Archives, you can see it. There's just little tiny dots. Medium-sized ones, and really large ones. So that would be an example of a really large colony. Now, form. Form is talking about the entire colony itself. What kind of form does it have? So we're going to have this thing called punctiform, which is very, very tiny colonies. And punctiform looks as though, as the name implies, they're punctuation, little dots, super little dots. Aside from that, anything that's bigger than just a super tiny little dot would be circular, so if it's completely round. Filamentous, it has kind of these fibers that are kind of growing out from it. And if you guys actually look back on this slide, these would be examples of filamentous colonies here. So they've got those little filaments. They can be irregular, so anything that's not round or isn't the other categories, we just say irregular, that's them. Rhizoid, which is kind of like lightning strikes coming out of the radius. Very uncommon. You guys won't see it. Don't worry about it. And the last one is spindle, which is these kind of almond-shaped colonies. Again, less common, probably won't see one. We can also talk about elevation. And these are actually really easy to differentiate. Flat is flat. I mean, it's almost perfectly level with the auger surface. You can see an example here, right there. Raised is basically the same thing, just a little bit higher and completely flat on top. Convex, a small little hill on the plate, so really tiny. Pulvinate, pulvinate is a much rounder, higher colony formation. And the last one is umbinate. Umbinate is basically pulvinate sitting on top of raised. So you've got a little raised platform and then just like a little nubbin on top right there. And that's what umbinate is. Any of these you guys can see in the bacteria that we grow, so keep an eye out for that. Margins has to do with the edge, the leading edge of the bacterial colony. So entire is just a smooth outside, really easy to do. Most bacteria kind of fit into that. You can have undulate, which is kind of a wavy edge, so basically not a solid edge. Now, people tend to get this one confused with lobate. Lobate has these little protrusions coming out, so little lobes, almost like fingers sticking out, trying to find new nutrients. So this would be an example of lobate. Eros. Boy, I wish I had found a better example of this. Eros is really hard to see in visualize on a picture, but if you guys, we used to call it serrated, and if you guys look at it, it looks kind of like the little teeth of a serrated knife, so little teeth edges all along the thing. And filamentous, curled, very uncommon. I couldn't find a picture of curled. That's how uncommon it is. I don't have one that makes curled. So there you go. So we'll talk about that. So the bacteria that we're using, the first one is Serratia. You can see it's got that nice bright red pigment, super bright, super stark. It likes to grow in starchy substances a lot. It's also found in things like well water. Fun fact, so it likes to grow on those starchy communion wafers, and some people would see that and think it was like, oh, it's the blood of Christ on the communion wafer. Well, it's actually Serratia because it produces a bright red pigment. And it does quite look like blood sometimes because it's very kind of moist in content. This is how it looks when it's growing in a broth. It's very turbid. It just kind of swims throughout the entire broth. It doesn't really care. It does move quite a bit, so you can see this in a semi-solid media. It kind of expands outward. And the last thing in this plate, and you can see this serrated edge right here, okay. Not great, but okay. So these little teeth. And that's the name Serratia. That's Serratia, serrated teeth. Does anybody know, anybody take a lot of Latin, know what marcescens means? No? Yes? Huh? It actually might be, yeah, that's actually a good point. So it talks about decay, and Mars is the Roman god of war. So that's actually not, it might actually come back to that etymologically speaking, but yeah, so it's decay, and typically they're talking about decay in the context of the pigment being degraded. But there you go. Okay, next one is E. coli, which we have messed around with a lot so far this semester. So you can see it growing on this nice plate. It's got these nice little white tannish colonies. When you grow it in a broth, it's kind of cloudy and turbid. On a slant, you can see it's kind of got those irregular undulate edges. In a deep, it kind of grows throughout the entire thing. This is very cloudy, semi-solid media. And since we're talking about this, which is the worst to spell bacterial name in the world, Escherichia coli, does anybody know where the name comes from? Like, so one of the names is Latin. Does anybody know what the Latin one is? Or have a wild guess? Huh? Okay, so coli, colon, that's what it comes from. So, E. coli is found in the guts of all mammals. The guy who discovered it in the guts of mammals and found that those bacteria were in the guts of all mammals. That's the name, coli. Escherichia is named after the guy, Thomas, let's see here, I gotta remember which part of his name stops at where. Thomas, Escherich, I believe is his name, Escherich. There you go. German guy. I can't say German names. There you go. So this is like a weird Latinization of his last name. So that actually has nothing to do with the bacterial name in Latin. It has all to do with the guy's last name. Okay, now hopefully you guys remember that and take these facts with you if you take nothing else from the class. Clean up stuff. Any stock cultures we have, make sure you guys put them on the autoclave after your group is finished. All plates and inoculated tubes should go in your lab-specific incubator, except for the one that I mentioned is not. Make sure that that one goes in the room temperature incubator. Please don't take the blue racks that are sitting at your table. Those are the communal ones, so don't take them because other people need them. On results day, of course, dispose of all the plates in the biohazard bags. Any tubes, put them in the appropriate cart. Lastly, disinfect your countertops and cell phone bags with Happicide. Of course, wash your hands and make sure you guys have a wonderful day. Before you guys leave, does anybody have any questions on anything we covered? No burning Latin? Yes. We will do the week after next. So we'll come back and do that later. Just, you can't do that all in one day, but we can do this all in one day. Yes? This is the 4th week. With 2 days of the 4th lab, so instead of reading the results on the 2nd day, we'll read it the week after that. Come here and I'll help you out with the lab archive stuff though.